Hello, everyone. Thank you so much for coming in and for joining us. We are just going to wait a couple minutes to get started, probably at about 12:02 or 12:03 Eastern Time. So thank you so much for being here. Can you see I just put it in presenter mode. Can you see that my screen now? Perfect. Hello, everyone. Thank you so much for joining. We're just going to wait about 2 minutes to get started while everyone is just filtering in for the event. Hi, everyone. Thanks for joining. Thank you. We'll get started in about a minute and a half. Also, this webinar is being recorded, so if you do want to watch it after, or if you want to share it with anyone after the event, you will get a link sent to the email that you registered. We'll just wait another minute for people to filter on, and then we will kick things off. All right, I think we can kick things off. Hello, everyone, and welcome to this Remember the Girls webinar on debunking X-linked dominant versus recessive inheritance. I'm Taylor Kane, founder and executive director of Remember the Girls, and I will be your moderator for today's event. Remember the Girls aims to break the stigma facing females impacted by X-linked conditions by providing them with tools to seek support, engage with research, and access family planning options, as well as by advocating for increased attention of medical professionals to the physical, emotional, and reproductive needs of this community. A few months ago, we learned of a newly published research paper calling for X-linked diseases to no longer be classified as X-linked dominant or X-linked recessive, adding to the growing body of research on this important topic. Today, we are joined by the leading author, Sanjana Basava, an upcoming junior at Northwestern University, studying biology and global health. Since 2022, she has been involved in research on X-linked inheritance, exploring how traditional models often fail to capture experiences of girls and women with genetic disorders. This project has not only extended her understanding of gene expression and chromosomal behavior, but also shown her how much medicine is still evolving. Outside of research, she's passionate about dance and serves as a captain of her college Bollywood fusion team. She loves working with children, whether through volunteering, tutoring, or teaching dance. Today, Sanjana is going to be sharing with us about her team's research findings in this paper. And if you do have any questions throughout the presentation, she prefers that you send those in the chat so we can address them during the presentation. She wants to make sure that there's nothing that is missed and you don't forget your questions. We will be also doing a Q&A at the end, but if there is something that comes up that she can dive a little bit more into, please feel free to send it in the chat so we can address it. Um, well, Sanjana, I am going to stop sharing and pass it to you to kick things off. Thank you so much for being here.
Yeah, thank you so much for having me here, Taylor. This is an amazing opportunity, and I love to share my research. And so let me just share my screen here.
Perfect.
Can everyone see this?
Yes,
Okay,
perfect.
awesome. So, like Taylor mentioned, I got involved in this research late 2021, early 2022, and I've been working on it ever since then. And so it's been a huge passion project of mine. And it's been really inspiring for me to see how medicine is constantly evolving and changing. And so that's like what drew me to this in the first place. And so yeah, it's really important to spread awareness and make sure that people are receiving the care that they deserve and the information that they deserve as well. And so this kind of introduces patterns of X-linked inheritance, which is like a new approach to the genome era. Um, okay, so just like a little bit of history of X-linked inheritance and some background. So early 20th century studies in the Drosophila laid the foundation. So this is basically the fruit fly. And so traits were classified as either X-linked dominant or X-linked recessive based on fruit fly observations conducted by Thomas Morgan in 1910. And so it was established that X-linked dominant inheritance means that a disorder or disease affects both sexes, and in X-linked recessive, a disorder only affects males. And so these models were applied to humans before mammalian sex chromosome biology was understood. And mammalian sex chromosome biology is often more complicated than Drosophila biology. And this is because of a difference in dosage compensation. So dosage compensation is a process that balances the amount of X-linked gene expression between males and females. So males have one X chromosome and females have two X chromosomes. And it's important that both of these sexes are producing similar levels of important functional proteins. So kind of analogy that I use to think about this is how if you think about a male and female being a factory, then males have one worker in the factory and females have two workers in the factory. How can I ensure that output for both of these factories is the same? And so both Drosophila and mammals have different methods to go about this. So in Drosophila, each X chromosome is expressed at 50% in each female individual cell. So this means that in the female factory, both workers are at 50%. However, in mammals, what happens is that one X is randomly inactivated within each female cell. And so this means that one worker is just not working. And so this process of X chromosome inactivation occurs in each individual cell. So we see kind of a mosaic in females, where if you look at figure B, we see that sometimes 50% of the cells are expressing one X chromosome and 50% of the cells are expressing another X chromosome. However, in figure C, we can also see that this leads to skewing of X chromosome inactivation, in which that 50-50 ratio isn't always a set in stone measure, and that sometimes 80% of cells can express one X chromosome, 20% of the other cells can express the other X chromosome. So this kind of leads to a fluidity and can lead to a spectrum of disease expression in females. And so this was found by Mary Lyon in 1961, and it just shows how the old standards and the old classifications were an oversimplification by Thomas Morgan that were set in 1910. And the current, so then the dominant and recessive categorizations are an oversimplification of how disease is presented in girls and women. And so kind of the limitations of these dominant and recessive labels, again, is just that real world human patterns of inheritance are so much more complex. And there's just a bunch of consequences that result from this historical oversimplification. So you've got misleading genetic counseling, underdiagnosis in females, biased literature, and very skewed data in which you only really see very sick women or very healthy women who are the mothers of sons that are affected. And we're kind of missing a spectrum of the phenotypes that are actually observed. And then we also see that educational materials for future physicians and healthcare professionals or academics are very outdated. Like even in my introductory genetics courses and even in medical school, X-linked recessive and X-linked dominant are still taught that way. And so it kind of leads to like improper education for future physicians and healthcare workers, leads to the same oversimplification occurring even through future caregiving and healthcare. And so we actually observed from our data collection, we analyzed 57 X-linked disorders, and we observed that female phenotypes exist on a spectrum of inheritance patterns, influenced by X chromosome inactivation skewing, like I talked about in the previous slide, cell autonomous or non-cell autonomous gene functions. So this means that if I have a product of a gene, which is usually protein, can that protein be shared with other cells? Or is it limited to that individual cell? So this can kind of relieve symptom expression sometimes when a healthy cell that can produce the proper protein can share that protein with a mutated cell that cannot express that protein. Female phenotypes also exist on a spectrum due to variant type and the severity of the mutation that actually occurs, and the severity of disease in that how important is this protein for human function? And so depending on how important it is, that can also influence how severe disease expression will be. And so we kind of categorized the X-linked disorders that we came across into 4 different groups based on patterns that we were observing. And so for our group 1 disorders, we saw that females were very rarely affected, so 0 to 10%. And skewed X inactivation protects females by inactivating the mutated X. So we saw very high skewed ratios, like 80-20 or above, where 80% of the cells or more of the female's body were expressing the correct protein, and the mutated X chromosome was preferentially inactivated, suggesting that this was a non-random process and some selection process was occurring to favor inactivating this mutated X chromosome. And so some examples of this are Menkes syndrome or Wiskott-Aldrich syndrome. Oh, okay. Sorry, I thought I was looking in the chat. I thought there were some questions, but
The question did pop up actually in the Q&A section. Do you want me to ask it to you now?
yes, sure.
Okay, so the question is, can non-cell autonomous expression go the other way? Could the affected gene give bad info to the non-affected gene, causing less function in the good gene and less effective protein production?
So what usually occurs is that it's not the gene that's being shared, it's the gene product. So genes and the DNA can't be swapped between cells. So if a malfunctional protein is made, the cell has machinery to degrade that and just kind of delete it. So it's usually just that it's not that the mutated protein is causing a symptom of expression, it's the lack of healthy protein that's causing the expression of disease. And so then the healthy protein can be shared because the mutated protein is usually often just deleted. So it's not the presence of the mutated gene, it's the lack of the healthy gene, if that makes sense.
That makes sense. Thank you.
Yeah, okay. So kind of going back to the group 1, so this is like very skewed X inactivation. And so for our group 2 genes, we noticed that this is when female expression of disease started getting higher, and there was a lot more variability in female expression of disease. And so we had to split this group into two subcategories. So we've got group 2A, and its defining characteristic was a functionally non-cell autonomous gene product. So like we were just talking about, healthy gene product or protein can move between different cells, and this offers a partial protection against disease. And so this can occur through three ways. Either a gene product is actively secreted outside of a cell. It is either it can diffuse across the cell membrane just by random chance, not like any active machinery secreting it out. Or this relates to disorders in muscle cells. So muscle cells often have multiple nuclei. So this means that one muscle cell can have multiple sets of DNA. And so this means that even if there's one set of DNA within the muscle cell that has a mutated X chromosome, there's probably also a set of DNA within the same muscle cell that has the healthy X chromosome. And so this means that the entire cell is still getting healthy gene product to an extent. And so this offers partial protection against disease, but not full protection against disease again, because we're still at that 50% capacity. You're not producing the same amount of healthy genes as you, and the same amount of healthy proteins as you should be. And so another interesting thing to think about, especially when you're looking at hemophilia, for example, is that females tend to have different types of symptoms, right? So when you look at heavy menstrual bleeding, this isn't a symptom that males have, but it's really important to recognize that it is a symptom of hemophilia, and it wasn't really considered until, the past 50 years. And so it's really important to recognize that even though, it's important to recognize that female expression of disease can be really different to male expression of disease, but that doesn't minimize that there is a female expression, and it's just as important to be studied. And so we look at group 2B genes, then moving on. This means, so we've got our cell autonomous genes. And so because that the gene product is limited to its individual cell, like the healthy cell can't help a mutated cell out, we see that female symptoms are slightly more severe. And so some examples of this are Fabry disease and CDKL5 related disorders. And just another note about Fabry disease, about a decade ago, it was thought that female expression of disease was around 30%. Now it's like upwards of 80%. So it just shows how this data is still evolving too, and it's important to keep updating it and keep adding more data points into the patterns that we observed, because a lot of the times, the data was just so poor. Next, moving on to group 3, these are very severe disorders in which males are often lethal. So females are often affected very severely, and males rarely survive. Either they survive during pregnancy or very soon after birth because of how severe this mutation is. And so some examples are Rett syndrome and incontinentia pigmenti. And these disorders seem like they're only affecting females, but it's just that males aren't able to live to show symptoms. It's that X chromosome inactivation, or the additional healthy X chromosome in females, instead of protecting females from expression of disease at all, it's actually preventing death in females that have this mutation. So this kind of shows how severity of disease also influences expression of disease. And then lastly, for our group 4 disorders, these are the actual female-limited disorders in which males with the same mutation are actually completely normal. And so this is a really interesting scenario, which is likely a result of cellular interference, in which signaling between cells is often lost. So this means that in males, even if they have a mutation, a mutated protein can interact with the mutated protein. And in females or males that don't have the mutation at all, healthy protein can interact with healthy protein. But with females, when you've got that heterozygous aspect, where you've got healthy and mutated proteins, the two of those cannot interact. And so an example of this is PCDH19 related developmental disorder. And it's showing how the mechanism for this disorder is that cell signaling is lost because healthy proteins and mutated proteins can't interact and send signals. And so this causes the disease expression in females. And so kind of overall, these were the patterns of X-linked inheritance that we observed. However, we expect these patterns, they may change, they may be more supported, but it's important for X chromosome inactivation research to be expanded because all of the patterns that we were looking at there was really only a pattern of skewing for group 1 disorders, but for groups 2, 3, and 4, it was really hard to find that data in females if it was present at all. And if it was present, it's important for X chromosome inactivation research to be done in multiple different tissues, right? So for example, for Rett syndrome, you've got that a lot of X chromosome inactivation studies were done in the blood, which is a type of tissue. However, Rett syndrome affects brain tissue. So it's really important to test X chromosome inactivation levels also in brain tissue. So it's important to kind of have all of those factors available and have that be expanded because it is super important when determining how female expression of disease can be affected. And we're also looking for more longitudinal and expansive studies of female expression of disease, kind of tracking over time, because a lot of the times female expression of disease, girls and women are affected a lot later in life than males and men are. And it's important to study this longitudinally and have these kind of studies available where you're tracking disease expression across a woman's entire life. Sorry, is there a chat? I think ALD is either in group 2 or group 3. I can look at my spreadsheet after the presentation is done and get back to you, but I can check that after the presentation if that's
And
okay.
it also in the paper, I think in the paper you you list the 57, do you list the 57 conditions that you looked at?
Yes. Yeah. So I think I can send that link or Taylor can send that link into the group chat as well. But there is a full table in how the 57 disorders are classified within the paper. But kind of going back to the presentation, so it's important that more data is available first. And then next, the terms X-linked dominant and X-linked recessive should just be completely retired. And these conditions should simply be described as X-linked with optional subgroup classification based on penetrance, which is how many females are affected, X chromosome inactivation patterns, and either the non-cell autonomous or the cell autonomous gene function. And so, in my classroom, they still teach it as X-linked dominant and X-linked recessive. And I spoke to my professor about this, and I was like, why is it taught this way when a lot of senior geneticists know that this isn't how X-linked inheritance really works? And he said that it was because he wanted to simplify teaching and understanding in students. But I honestly think that X-linked is a lot more simple than X-linked dominant and X-linked recessive. So it's really important that these terms are just removed from the vocabulary when discussing X-linked inheritance, because it can lead to a lot of future misconceptions about how X-linked inheritance works, especially when these terms are taught throughout medical school as well. It's important that future physicians know how X-linked inheritance works. And kind of going off of that, it's really important to update educational materials and definitions based on these dominant or recessive concepts within X-linked inheritance should be removed from textbooks, curricula, and scientific literature. I know Taylor's working on a really cool project called Rare Rewritten, and it's about, kind of spreading awareness that these terms and concepts should be removed from different websites and different informational areas to ensure that misconceptions about X-linked inheritance don't persist. And it's just really important that people are able to fully understand. And so just having that information is just really important, in my opinion. And then that's kind of it. So, questions.
Great. Thank you so much, Sanjana. That was incredibly informative and really helps, for so long, I think within our communities, we've been saying, these terms do not make sense, the condition that I'm impacted by, adrenoleukodystrophy, is another one that's often referred to as X-linked recessive, despite over 90% of women having symptoms. And, I think now having this data and these different categorizations can really help us in Remember the Girls with, pushing to do away with these X-linked dominant versus X-linked recessive and showing that, it's not either or. So thank you so much for this research. And, I have a few questions prepared.
Mhm.
But please, if anyone has any other questions, to send them in the chat or send them in the Q&A. I see Shelley's question, but I'll actually get to that at the very end when I talk about our Rare Rewritten campaign that you mentioned. But I guess my first question is, you mentioned how even though a lot of experts, know that X-linked dominant and X-linked recessive are not, proper or effective categories, why do you think that medical literature and, teachings in medical school have continued to learn these? Do you think it's really because they think it's just easier, or um, what do you think about that?
I think like from a historical perspective too, that there was so much establishment of X-linked recessive, X-linked dominant, and how I think genetics is often like a forgotten subject in medicine sometimes where it's not updated as frequently or as often. And a lot of these misunderstandings also arise from like kind of a lack of data. If you kind of look at it, X-linked X chromosome inactivation is a fairly new concept. It was only found in 1960 when a lot of these genetic studies were done in the 1800s, early 1900s. And so I think it's like a combination of it's a simple way to understand it. Um, Women's disease expression was often like very dismissed historically. And I think even when you think about like a historical perspective of what hysteria was and how women were often like affected with such severe diseases, but it was just kind of dismissed as, oh, she's crazy or she's mental. And I think obviously not anymore now to an extent, but just that kind of historical lack of data, I think, is what has prevented a lot of understanding to be there. But also, yeah, I don't know. I think it's just, in my opinion, it's just like the lack of data sometimes is what kind of makes it a lot harder. And as more data becomes available, more and more people will kind of see this and agree.
That makes total sense. And that leads into my other question. So, when we first met, and we also spoke with Dr. Dobbins, who's done a lot of research on this topic and is one of the co-authors, you both were talking about how, a lot of the data in X-linked diseases is not comprehensive because a lot of, the women in studies are probans, or that was a new term to me, but I think that's, the mother of the son who was diagnosed. So, she is quote unquote the carrier and, may not have symptoms herself. Obviously, there's a good chance she will, but typically, they're not being diagnosed through symptoms. So they're going to be less severe
Mhm.
than, say, a woman presenting to a neurologist with severe symptoms who might more likely be diagnosed with ALD outright, which is starting to happen more than it did,
Mhm.
before. But can you talk about why it's so essential to have not just probans or mothers of affected sons in research, and why we really need the full spectrum of women and girls?
Yeah, so like even when we were looking at the data points that we wanted, so like we saw that either it was mothers of affected sons coming to the attention of healthcare workers, or it was girls with really, really severe symptoms that were coming to the attention, and like the in between was kind of lost. Let me look. Let me unshare my screen once, and there was a specific disorder that we did this for. I think it was ARX, and we found that when you got rid of the mothers of affected sons and you were only looking at female relatives of this son, for example, that were like aunts or siblings of a grandmother or anyone that wasn't in the direct maternal line of that son, you found that female expression of disease, the penetrance or the, I'm losing the word for it, like the percentage of the women affected went up a ton compared to if you included the mothers of the affected sons. So it's really important to see that a lot of the data is just so heavily skewed one way that we really aren't able to tell how many women are actually affected with these disorders, as because a lot of the symptoms can be mild or non-clinic, um, like they don't affect a woman's life to the same extent as they affect a male's life. And so a lot of these symptoms are just dismissed or just kind of brushed under the rug. Oh, it doesn't really matter. Like, and then a lot of women kind of miss having that chance to get a proper diagnosis. And so we lose a lot of the females in the middle on how they're affected by disease. And so I think it's really important that if an affected son comes into a clinic, that a full proper family history is done and all of the females in the family are tested, because like they can present symptoms in the future and it's really important that they have that information. So this is just an example of how genetic counseling can be done in the future to kind of account for female expression of disease.
That makes total sense. And, I think it can even be women themselves aren't
Mhm.
aware that things they're experiencing are symptoms because they might know about the male expression of the disease and they can look at their sons or their male family members and think, oh, I don't have, I'm still able to walk or I'm still able to do this. So I must not have symptoms. But that's why, as you mentioned, we really need to look and understand longitudinally what these diseases look like in females. And then for these women to also know themselves, what the symptoms are in women and how those might differ or look different than in men. Another question that we got, do you think people still use those terms, especially due to the ease of use in pedigrees? I remember being taught that if more males are shown as affected, it is X-linked recessive and dominant if there's an equal number of males and females affected.
You know, I don't understand. I mean, with pedigrees, obviously, in like problem solving scenarios, yes, it's simpler to teach it this way, but that's often not how it presents in like actual families. You see that women are affected to a certain degree, obviously, not as severely as males. But even in X-linked dominant disorders, males were often a lot more severely affected than females. So I don't know. I don't think pedigrees were able to really establish the expression of female disease. Um, but yeah, I think X-linked dominant is kind of confusing because it implies that females and males are affected exactly the same and to the exact same severity. But oftentimes we did see that most of the time, actually, like honestly, all of the time, except maybe one disorder, we observed that males were affected to a lot more severe degree than women. So then X-linked dominant becomes confusing, and then X-linked recessive becomes confusing because that implies that women aren't affected at all. And then women are showing some cases of a disease expression. And so classroom pedigrees can be like a super duper oversimplification of how female expression of disease actually works. And so that's why I don't think they're a good way of teaching X-linked inheritance, because I think with pedigrees especially, it's like solid or blank or solid or unfilled. And so that implies that there's no in between for symptoms. And so I think that's how pedigrees can kind of oversimplify how disease expression works. But I think I do agree that maybe it is taught that way because it is easier to learn it through pedigrees. I think that is a good point. And so I just don't think the pedigrees are accurate in showing how female expression of disease actually works.
Thank you for the response. Um, Another question that we have, do you know of anyone else researching this, or basically, what are the next steps in this research?
You know, there is a lot of research being done in X-linked inheritance. I think Dr. Barbara Midgen is a great example. She's retired now, but a lot of her papers established a really good foundation for this field and how X chromosome inactivation can really affect how female expression of disease happens. There's also experts within specific diseases that are studying, like that are hopefully creating, these longitudinal studies that I was talking about, not just looking at the whole, but looking at in each specific disease. Like, for example, Fabry disease has had a lot of research on female expression disease come out. And so there's a bunch of research being done within each individual disorder, which is super awesome and important as well. And so I think, yeah, the field is expanding and it's really good to know. And it's good to know that people are noticing that something is off and something is wrong and that women are affected a lot more than we realize. So it is a growing field, yes.
That's great to hear, especially since, of course, these labels have real world implications. For example, a lot of the conditions in your study, I'll use ALD and Fabry. I know that they've long both been considered X-linked recessive. And for example, on the Medline Plus website, which is run by the NIH, it says X-linked recessive diseases cannot present in women because a heterozygote would need that it would not be possible unless that female has two mutations.
Mhm.
On both of her X chromosomes. And as we know, these women in our group, they have one mutation and they're heterozygous, yet a very high percentage of them are having symptoms. So it's very clear that on some of the most prominent medical websites, there's very misleading information, and that's not only harmful to, the clinicians who might see that and see a patient, but also for the women themselves who maybe are told you have an X-linked recessive diagnosis, and then they look and see, oh, well, it's not possible for me to have symptoms then. So then,
Yeah.
they could be misdiagnosed with something else if they're seeking treatment for their symptoms, because yeah, they're being led this information that is very outdated. But what do you think that students studying medicine and new clinicians and genetic counselors can do to help with this existing disparity?
I think it goes down to the teaching and what's taught in medical school. I know that this kind of niche topic of X-linked inheritance is treated as niche. So, like it's not really thought of as really important information to learn unless you are a very subspecialized genetic counselor or a geneticist or a physician working very closely in genetics. And I think it's really important for even normal physicians like working in pediatrics or internal medicine to kind of know these terms and know that that's not how it actually works, because how healthcare usually progresses is like you go to your primary care physician and then they can recommend you to another physician that's more specialized. And so it's important for these primary care physicians to also know that X-linked inheritance works to affect females as well. And so then they know who to direct them to. And I think it's really important that this is kind of taught starting in med school itself so that any future physician will know that yes, this could be a genetics this could be related to genetics, and it's super important for that awareness to be there so that misdiagnoses don't continually occur.
Definitely. Does anyone have any final questions? We have time for a couple more questions before we move on.
I do want to, I think Trina said that she's working hard on changing the terminology from manifesting carrier or affected carrier. And I think that's so important. I think the term carrier itself is so, it implies that the carrier is not affected at all. And that is often not the case at all. And so I think that's really awesome because I think like it's so important that I think just terminology changing is so important because then it makes sure that misconceptions don't arise and that misunderstandings don't persist. And starting with the terminology will really help raise awareness because just sticking a label of carrier on someone just means like, oh, she's not a problem we have to deal with because she's not really affected. But getting rid of like those labels and those incorrect labels will, I think, hopefully do a lot in spreading awareness and ensuring that people receive the care that they deserve.
Definitely agreed. And it is interesting how like affected carrier, like those two words are opposite of each other. So, yeah, it doesn't really make sense, especially in, these conditions in groups 2, 3, and 4, especially where it's like a very large percentage. And I think in the study you did, most conditions were in group two, 2 or 3.
Mhm.
So, most of these X-linked conditions, females are affected,
Yeah,
and two, a decently high percentage of females are affected. So the term is just doesn't make sense.
yeah.
Um, we got another question. Is there any concern about stigmatizing women with X-linked conditions, like adding weight or anxiety for reproductive purposes?
This is a really good question, actually. I mean, I think the more information available can actually reduce the stigma. I think there's like, I think having all the information available will, I mean, genetic counseling isn't there to kind of create stigmas, but rather give people the information that they should have and want before going about family planning. But I guess I didn't really like,
Yeah, I can like talk about that just from the
yeah,
perspective of a woman with an X-linked condition. I think that there is already so much negative stigma around the term carrier. Whether I think it even goes back, you talked about the gender bias in medicine and how women's um conditions were long misunderstood. But I also saw a paper once about, the term carrier and going back to Typhoid Mary, kind of random name throwing, but she is often thought of as like the OG carrier. Obviously, that's an infectious disease, but she was this woman who went around and affected other people, but was not symptomatic herself. And I think that women in our community who are just told, oh, you're just a carrier. So you're not affected, but you pass it down and you cause harm to other people, basically. There's this negative stigma associated with it already. And because of that, a lot of women in our community feel this extra weight of guilt by thinking, oh, I'm not affected, but I put this weight on others. And we know it's true that most women actually are affected. Of course, some do remain asymptomatic, but I think that women understanding their risk for symptoms and also, understanding that when you look on for males, it was actually Shelly. She's here, or she was here on this call, but she had brought up how she used to wear a shirt at hemophilia conferences that said, "My dad is a hemophilia carrier," to be like ironic because her dad passed it to her. My dad passed it to me, but no one would ever call him a carrier of ALD, even though he didn't experience symptoms till he was in his 40s. But they would never call him a carrier because he's a male, even though genetically he passed the condition to me. So I think I can understand where this question is coming from, but I think that there's already a lot of stigma with the term carrier, and that, the more information and the better we can, classify these conditions to make sure that women get the care that they need, the better. But that's my individual perspective, not on behalf of Remember the Girls, but yeah, I really appreciate that question because it is always important to remember, how these things will impact the community. All right. Let's see. Okay, answered this. Okay, so I think that is it for questions. So I'm going to share my screen just one final time, and also address Shelly's question, just about what we can do basically with this information. So, Sanjana, thank you so much for your research. The fact that you are an undergrad and that you're already a leading author on this paper, I can't even imagine all you're going to accomplish, going to medical school and beyond. So thank you for sharing this with our community. And before we end today's webinar, I want to talk about the campaign that we teased a little bit, called Rare Rewritten. So this is going to be launched this month. And as part of this campaign, Remember the Girls is going to be sending letters to numerous medical websites, asking them to update their information on X-linked inheritance and how X-linked conditions impact females. In this letter, which will be shown to the public on our landing page and our website, we reference various research studies, including the one that Sanjana is an author on, that we talked about today. We reference those research studies and, really back up these points that we're making with data, um, to ask these medical websites to update the information accordingly. So we'll be sharing more details soon. Make sure you keep an eye on our social media accounts, and also subscribe to our newsletter, all of which you can do through our website, rememberthegirls.org. But I think it's definitely time, that the medical community and, the patient community, work together on, getting some of this information corrected because, we have the data to support it, and we need, this to be correct both for the women in our community and also any clinicians, who might access information as well. So thank you all so much for coming. And thank you again, Sanjana. You were amazing.
Thank you so much for having me. This was awesome. And I love sharing and talking about this topic. So this was really awesome.
Great. Thank you, everyone. Okay, I'm going to stop the recording now.